Good day, this is Professor Will, CBMD PhD. Today's Tuesday, May 22nd, 2018. It is 5:53 a.m. Eastern Standard Time. Continuation of science news. Mice regrow brain tissue after stroke with bioengineered gel. Replacement neurons, blood vessels fill in stroke cavity gel provides scaffolding. In a first of its kind finding, a new stroke healing gel will help regrow neurons and blood vessels in mice with stroke-damaged brains. UCLA researchers report in the May 21st issue of Nature Materials. We tested this in laboratory mice to determine if it would repair the brain in the model of stroke and lead to recovery. Thomas Carmichael, professor of chair of neurology, UCLA, this study indicated that new brain tissue can be regenerated in what was previously just an inactive brain scar after stroke. The brain has a limited capacity for recovery after stroke. Other diseases, unlike some other organs in the body, such as the liver or skin, the brain does not regenerate new connections, blood vessels, or new tissue structures. Tissues that dies in the brain from stroke is absorbed, leaving a cavity devoid of blood vessels. Neurons or axons, the thin nerve fibers that project from neurons. To see if healthy tissue surrounding the cavity could be coaxed into healing the stroke injury, scientists engineered a gel to inject into the stroke cavity that thickens to mimic the properties of brain tissue, creating a scaffolding for new growth. The gel is infused with molecules that stimulate blood vessel growth and suppress inflammation, since inflammation results in scars and impedes regrowth of functional tissue. After 16 weeks, stroke cavities in mice contain regenerated brain tissue, including new neural networks, a result that had not been seen before. The mice with new neurons showed improved motor behavior, though the exact mechanism wasn't clear. The new axons could actually be working, or the new tissue could be improving the performance of the surrounding unharmed brain tissue. The gel was eventually absorbed by the body, leaving behind only new tissue. This research was designed to explore recovery in acute stroke or the period immediately following stroke. In mice, that is 5 days. In humans, that is 2 months. Next, scientists are determining if brain tissue can be regenerated in mice long after the stroke injury. More than 6 million Americans are living with the long-term outcomes of stroke, known as chronic stroke. Good day, this is Professor Will, CBMD PhD. Today's Tuesday, May 22nd, 2018. It is 6:03 a.m. Eastern Standard Time. Continuation of science news. Hotter bodies fight infections and tumors better. Researchers show how. The hotter our body's temperature, the more our bodies speed up the key defense system that fights against tumors, wounds, or infections. The researchers have demonstrated that small rises in temperature, such as during a fever, speed up the speed of a cellular clock that controls the response of infections. And this new understanding could lead to more effective and fast-working drugs which target a key protein involved in this process. Biologists found that inflammatory signals activate nuclear factor kappa B transpose, NF-KB proteins to start a clock ticking in which NF-KB proteins move backwards and forwards into and out of the cell nucleus, where they switch genes on and off. This allows cells to respond to the tumor, wound, or infection. When NF-KB is uncontrolled, it is associated with inflammatory disease such as Crohn's disease, paralysis, and rheumatoid arthritis. At a body temperature of 34 degrees, the NF-KB clock shows down at higher temperatures than the normal 37-degree body temperature, such as in fever 40 degrees. The NF-KB clock speeds up. Mathematicians at the University of Warwick's System Biology Center calculated how temperatures increase make the cycle speed up. They predicted that a protein called A20, which is essential to avoid inflammatory disease, might be critically involved in this process. The experimentalists then removed A20 from cells and found that the NF-KB clock lost its sensitivity to an increase in temperature. Scientists explain that in normal life, the 24-hour body clock controls small 1.5 degree changes in body temperature. He commented that the lower body temperature during sleep might provide a fascinating explanation into how shift work, jet lag, or sleep disorders cause increased inflammatory disease. While the activities of many NF-KB control genes were not affected by temperature, a key group of genes showed altered profiles at different temperatures. These temperature-sensitive genes included the key inflammatory regulators and controllers of cell communications that can alter cell responses. This study shows that temperature changes inflammation in cells and tissues in a biologically organized way and suggests that new drugs might more precisely change the inflammatory response by targeting the A20 protein. The professor, Mike White, lead biologist at the University of Manchester, said the study provides a possible explanation of how both environmental and body temperature affects our health. We have known for some time that influenza and cold epidemics tend to be worse in the winter when temperatures are cooler. Also, mice living at higher temperatures suffer less from inflammation and cancer. These changes may now be explained by altered immune responses at different temperatures. Good day, this is Professor Will, CBMD, PhD. Today's Tuesday, May 22nd, 2018. It is 6:14 a.m. Eastern Standard Time. Science News. New 3D printer can create complex biological tissues. Device could help advance regenerative medicine. Date, May 21st, 2018. Source, UCLA Samuel School of Engineering. Summary, scientists have developed a specially adapted 3D printer to build therapeutical biomaterials from multiple materials. The advance could be a step toward on-demand printing of complex artificial tissues for use of transplants and other surgeries. Tissues are wonderfully complex structures, so to engineer artificial versions of them that function properly, we have to recreate their complexity. The technique uses a light-based process called stereolithography and takes advantage of customized 3D printer designed by scientists that has two key components. The first is a custom-built microfluidic chip, a small flat platform similar in size to a computer chip with multiple inlets. Each prints a different material. The other component is a digital micromirror, an array of more than 1 million tiny mirrors that each moves independently. The researchers use different types of hydrogels, materials that after passing through the printer form scaffolds for tissue to grow into. The micromirrors direct light onto the printing surface, and the illuminated areas indicate the outline of a 3D object that being printed. The light into triggers molecules bonds to form in the materials, which cause the gels to firm into solid material. As a 3D object is printed, the mirror array changes the light pattern and indicate the shape of each new layer. The process is the first to use multiple materials for automated stereolithic bioprinting, an advance over conventional stereolithic bioprinting, which has only one type of material, while the demonstration device uses for 4 types of bioinks. The study authors write that the process could accommodate as many inks as needed. Researchers first used the process to make simple shapes such as pyramids, and they made complex 3D structures that mimicked parts of muscle tissue and muscle skeleton connective tissues. They also printed shapes mimicking tumors with networks of blood vessels, which could be used as biological models to study cancers. They tested the printed structures by implanting them in rats. The structures were not rejected. Good day, this is Professor Will, CBMD, PhD. Today is Tuesday, May 22nd, 2018. It is 6:33 a.m. Eastern Standard Time. Daily egg consumption may reduce cardiovascular disease. Having an egg a day could reduce the risk of stroke by 26%. Date, May 21st, 2018. Source, BMJ. Summary, people who consume an egg a day could significantly reduce their risk of cardiovascular disease compared with eating no egg, suggests a new study. Cardiovascular disease, CVD, is the leading cause of death and disability worldwide, including China, mostly due to ischemic heart disease and stroke, including both hemorrhagic and ischemic stroke. Unlike ischemic heart disease, which is the leading cause of premature death in most Western countries, stroke is the most responsible cause in China, followed by heart disease. Although ischemic stroke accounted for the majority of strokes, the proportion of hemorrhagic stroke in China is still higher than that in income countries. Eggs are a prominent source of dietary cholesterol, but they also contain high-quality protein, many vitamins, and bioactive components such as phospholipids and carotenoids. Previously, studies looking at associates between eating eggs and impact on health have been inconsistent, and most of them found insignificant associations between egg consumption and coronary heart disease or stroke. They used data from the China Biobank (CKB) study, an ongoing prospective study of around 5,512,891 adults aged 30 to 79 from 10 different geographic areas in China. The participants were recruited between 2004 and 2008 and were asked about the frequency of their egg consumption. They were followed up to determine their morbidity and mortality. For the new study, the researchers focused on 416,213 participants who were free of prior cancer, cardiovascular disease, CVD, and diabetes. From that group, a median follow-up of 8.9 years, of total of 83,977 cases of CVD and 9,985 CVD deaths were documented, as well as 5,103 major coronary events. At the start of the study period, 13.1% of participants reported daily consumption usual amount 0.76 egg per day, and 9.1% reported never or very rare consumption usual amount 0.29 egg a day of eggs. Analysis of the results showed that compared with people not consuming eggs, daily consumption was associated with a lower risk of CVD overall. And in addition, there was 12% reduction in risk ischemic heart disease observed for people consuming eggs daily, estimated about 5.32 eggs a week, when compared with the never rarely consumption category 2.3 eggs a week. This was the observational study, so no firm conclusions can be drawn about cause and effect. But the authors said their study had a large sample size and took into account established and potential risk factors for CVD. The authors concluded the present study finds that there is an association between moderate levels of egg consumption up to 1 egg a day and a lower cardiac event rate. Our findings contribute scientific evidence to dietary guidelines with regard to egg consumption for a healthy Chinese adult. Good day, this is Professor Will, CBMD, PhD. Today is Wednesday, May 23rd, 2018. It is 4:56 a.m. Eastern Standard Time. Spooky Action, a distant researchers developed module for quantum repeater. Date, May 22nd, 2018. Source, Saarland University. Summary, physicists have succeeded in entangling a single atom with a single photon in the telecom wavelength range. The constituents a basic building block for a transmission of quantum information over long distance with low loss. And communication using quantum states offers ultimate security because eavesdropping attempts perturb the signal and would therefore not remain undetected. For the same reason, though long-distance transmission of that information is difficult, in classical telecommunications, the increasing attenuation of signal is counteracted by measuring, amplifying, and resending it in so-called repeater stations, but this turns out to be as detrimental to the quantum information as an eavesdropper. And therefore, a different principle has to be used. The quantum repeater. Here, quantum entanglement is established over a short distance and then propagated to longer separations. Quantum entanglement between two particles means that their common state is precisely defined, although when one measures the individual states of the particles, the results are random and unpredictable. A possible realization is to entangle a single atom with a photon that emits. For the wavelength of 854 nanometers where atom-photon entanglement is created, however, no low-loss optical fibers for long-distance transmission exist. Instead, one would like to transmit the photons in one of the so-called telecom bands, 1315-60 nanometers. The technology for converting the photons into this regime, the quantum frequency converter, has been developed. Together, the two groups have now demonstrated that after quantum frequency conversion, the telecom photon is still entangled with the atom that emitted the original photon, and that the high quality of entanglement is maintained. One of the fascinating aspects of the work is that the entangled quantum state of two microscopic particles, a single atom and a single telecom photon, extends over several floors of the physics building of the university. Illustration of quantum entanglement. The state of individual quantum bit, an atom with two energy states of its electron, or a photon with two directions of its polarization, may be visualized as a point on the surface of a sphere. Measurement of that state provides an unpredictable result anywhere on the surface. The other qubit that is entangled with the first one will, however, always be found in the opposite point on the sphere. The correlation may also exist over large distances. Einstein called this phenomenon spooky action at a distance. It belongs to the non-intuitive peculiarities of quantum mechanics, but it has been confirmed in many experiments. Good day, this is Professor Will, CBMD, PhD. Today is Wednesday, May 23rd, 2018. It is 5:10 a.m. Eastern Standard Time. Science News. Early life trauma and been associated with reduced levels of sperm microRNAs. In mice, the same changes in sperm microRNAs occur and are transmitted to the next generation. Date, May 22nd, 2018. Source, Tufts University Health Science Campus. Summary, exposure to early life trauma can elevate risks for poor physical and mental health in individuals and their children. A new epigenetics study in both men and mice posit that some of the vulnerability in children may derive from stress-associated reductions in microRNAs in their father's sperm. And A new epigenetic study raises the possibility that the same is true in humans. It shows for the first time that the levels of the same two sperm microRNAs change in both men and mice exposed to early life stress. In mice, the negative effects of stress are transmitted to offspring. The study raises the possibility that some of the vulnerability of children is due to Lamarckian-type inheritance derived from their parents' experiences. According to Chief Trends Research Brief published in 2014, a remarkably high percentage, 10% of the population report scores are above this cutoff. MicroRNAs constitute a newly appreciated type of gene regulator where each microRNA controls a distinct set of genes. Until recently, sperm from fathers were thought to contribute only DNA to the mother's egg upon fertilization. But new data in mice indicate that sperm also contribute microRNAs that influence the next generation. Sperm microRNAs expression in humans is known to be affected by environmental factors such as smoking and obesity, but no human study to date has documented the effects of stress. A new study found that among 28 Caucasian male volunteers, the expression of highly related microRNAs, MIR449 and MIR34, were inversely proportional to the men's ACE scores. Men with the most extensive early abuse highest ACE scores had as much as 300-fold reduction in the two sperm microRNAs compared to men with the least abuse. The idea that these changes can affect the next generation is supported by additional findings in this study. The same sperm miRNA changes that take place in men with high ACE scores also occur in mice exposed to early life social instability stress, which lab has shown previously leads to anxiety and sociability effects in female offspring of stressed males for at least 3 generations. These two sets of microRNAs are known to work together in mice to allow proper development of brain and sperm. In humans, microRNA 34 has been implicated in promoting early embryo development. The mouse study showed that the decline in these sperm microRNA levels is transmitted to the next generation, and when these embryos mature, these microRNAs are also reduced in sperm of their male offspring to pass on stress behaviors to their female offspring. This is the first study to show that stress is associated with altered levels of sperm microRNAs in humans. We are currently setting up a new large study in men, and additional experiments in mice that could yield further support for the idea that changes in these sperm microRNAs do, in fact, contribute to an elevation of stress-related disorders across generations. Looking to the future, we may be able to figure out a way to restore the low microRNA levels found in men exposed to extreme trauma, because epigenetic changes such as stress-induced decreases in sperm microRNA expression are reversible, unlike genetic changes that alter the DNA sequence. For example, obesity has been shown to alter specific sperm microRNA levels in men, while bariatric surgery and subsequent weight loss can reverse the changes. In addition, Isabel Mansouri lab has reversed some of the negative effects of stress in mice across generations by exposing mice to an enriched environment that involves extensive social interactions, exercise, and opportunities to explore their surroundings. In addition to focusing on the potential transgenerational effects of stress, there is a growing appreciation that physicians should collect information on childhood trauma for the sake of the patients who are experiencing the early trauma. This is because childhood abuse, trauma, and dysfunction adds to the risk of future physical and psychiatric maladies. The significant exposure to abusive and dysfunctional families is remarkably common. Moreover, sensitivity to PTSD has been shown to correlate with ACE score, implying the ACE questionnaire could be used as a screening tool to identify people who should take extra precaution to avoid potentially traumatic experiences. However, some people may not answer the ACE survey accurately due to inaccurate recall or because of the sensitive nature of many of the questions, particularly in settings that do not allow anonymity and or where their answers could affect their future. Thus, discovery of unbiased markers for early trauma, like specific sperm microRNAs content, could complement ACE surveys in some clinical settings to bolster preventive medicine. The authors note that the relatively small sample size limits their ability to more deeply explore the association between ACE scores and microRNA expression. In addition, a longitudinal study with information on behavioral and psychological factors throughout adulthood, with repeated measurements of sperm microRNA content, could allow for further exploration on the effect of cumulative exposure to childhood trauma on microRNA. And new tech may make prosthetic hands easier for patients to use. Date May 22, 2018. Source North Carolina State University. Summary: researchers may have developed a new technology for decoding neuromuscular signals to control powered prosthetic wrists and hands. The work relies on computer models that closely mimic the behavior of the natural structures in the forearm, wrist, and hand. The technology could also be used to develop new computer interface devices for applications such as gaming and computer-aided design. The technology has worked well in early testing but has not yet entered clinical trials, making it years away from commercial availability. The work was led by researchers in Joint Biomedical Engineering Program at North Carolina State University, University of North Carolina at Chapel Hill. Current state-of-the-art prosthetics rely on machine learning to create a pattern recognition approach to prosthetics control. This approach requires users to teach the device to recognize specific patterns of muscle activity and translate them into commands such as opening or closing prosthetic hand. Pattern recognition control requires patients to go through a lengthy process of training their prosthesis. The process can be both tedious and time-consuming. He wanted to focus on what we already know about the human body. Who is a senior author of the paper on the work? This is not only more intuitive for users, it is also more reliable and practical. That's because every time you change your posture, your neuromuscular signals for generating the same hand-wrist motion change. So relying solely on machine learning means teaching the device to do the same thing multiple times. Once for each different posture. Once for when you are sweaty versus when you are not, and so on. Our approach bypasses most of that. Instead, the researchers developed a user-genetic musculoskeletal model. The researchers placed electromyography sensors on the forearms of 6 able-bodied volunteers, tracking exactly which neuromuscular signals were sent when they performed various actions with their wrists and hands. This data was then used to create the genetic model, which translated those neuromuscular signals into commands that manipulate a powered prosthetic. When someone loses a hand, their brain is networked as if the hand is still there. So if someone wants to pick up a glass of water, the brain still sends those signals to the forearm. We use sensors to pick up those signals and then convey that data to a computer, where it is fed into a virtual musculoskeletal model. The model takes the place of the muscles, joints, and bones, calculating the movements that would take place if the hand and wrist were still whole. It then conveys that data to the prosthetic wrist and hand, which perform the relevant movements in a coordinate way and in real time, more closely resembling fluid natural motion. By incorporating our knowledge of the biological process behind generating movement, we were able to produce a novel neural interface for prosthetics that is genetic to multiple users, including an amputee in the study, and is reliable across different arm postures. And the researchers think the potential applications are not limited to prosthetic devices. This could be used to develop computer interface devices for able-bodied people as well, such as devices for gameplay or for manipulating objects in CAD programs. In preliminary testing, both able-bodied and amputee volunteers were able to use the model-controlled interface to perform all the required hand and wrist motions, despite having very little training. We are currently seeking volunteers who have transradial amputations to help us with further testing of the model to perform activities of daily living. We want to get additional feedback from users before moving ahead with clinical trials. To be clear, we are still years away from having this become commercially available to clinical use, and it is difficult to predict potential costs since our work is focused on the software and the bulk of costs for amputees would be in the hardware that actually runs the program. However, the model is compatible with available prosthetic devices. The researchers are also exploring the idea of incorporating machine learning into a generic musculoskeletal model. Our model makes prosthetic use more intuitive and reliable, but machine learning could allow users to gain more nuanced control by allowing the program to learn each person's daily needs and preferences and better adapt to a specific user in the long term. Good day, this is Professor Will, CBMD, PhD. Today's Saturday, May 26, 2018. It is 7:14 a.m. Eastern Standard Time. Social media posts may signal whether a protest will become violent. Date, May 23, 2018. Source, University of Southern California. Summary: scientists have developed a model for detecting moralized language based on prior deep learning framework that can reliably identify text that reflects moral concerns about an issue. The research also finds that people are more likely to condone using violence to defend their beliefs when they think others share their moral values. And the researchers also found that people are more likely to endorse violence when they moralize the issue that they are protesting, and when they believe that others in their social network moralize that issue too. Utilizing a deep neural network and advanced machine learning technique to detect moralized language, the scientists analyzed 18 million tweets posted during the 2015 Baltimore protests for a Gray 25 who died as police took him to jail. In fact, tweets containing moral rhetoric nearly doubled on days when clashes among protesters and police became violent. The study was published on May 23rd in Nature Human Behavior. Social media is a barometer for activism. Social media sites such as Twitter have become a significant platform for activism and a source for data on human behavior, which is why scientists utilize them for research. Recent years of movements tied to social media include the March for Our Lives effort to seek gun control, the Me Too movement against sexual assault and harassment, and Black Lives Matter, a campaign against systematic racism which began in 2014 after the police-involved shooting death of Michael Brown, 19, in Ferguson, MON. A more violent example is the Arab Spring Revolution, which began in Tunisia in the late 2010 and set off protests in many other countries, including Egypt and Libya, that forced changes in their leadership. In Syria, clashes escalated into a war that has killed hundreds of thousands of people and displaced a multitude of refugees. Detecting moralization online. The scientists developed a model for detecting moralized language based on a prior deep learning framework that can reliably identify text that evokes moral concerns associated with different types of moral values and their opposites. The values, as defined by a moral foundations theory, are focused on care, harm, fairness, cheating, loyalty, betrayal, authority, subversion, and purity, degradation. Why more protests are violent? Social media data help us illustrate real-world social dynamics and test hypotheses in situ. However, as with all observational data, it can be difficult to establish the statistical and experimental control that is necessary for drawing reliable conclusions. To make up for this, the scientists conducted a series of controlled behavioral studies which, with more than 200 people, how much they agreed or disagreed with statements about the use of violence against the far protesters after they had read a paragraph about the 2017 clashes over the removal of Confederate monuments. The more certain people were that many others in their network shared their values and views, the more willing they were to consider the use of violence against their perceived opponents, the scientists found. And good day, this is Professor Will C., BMD, PhD. Today's Saturday, May 26, 2018. It is 7:31 a.m. Eastern Standard Time. Ingestible bacteria on a chip could help diagnose disease. Ultra-low power sensors carrying genetically engineered bacteria can detect gastric bleeding. Date, May 24, 2018. Source, Massachusetts Institute of Technology. Summary: researchers have built an ingestible sensor equipped with genetically engineered bacteria that can diagnose bleeding in the stomach or other gastrointestinal problems. MIT researchers have built an ingestible sensor equipped with genetically engineered bacteria that can diagnose bleeding in the stomach or other gastrointestinal problems. This bacteria-on-a-chip approach combines sensors made from living cells with ultra-low power electronics that convert the bacterial response to the wireless signal that can be read by a smartphone. And by combining engineered biological sensors together with low-power wireless electronics, we can detect biological signals in the body and in near real time, enabling new diagnostic capabilities for human health applications. In the new study appearing in the May 24th online edition of Science, the researchers created sensors that responded to home a component of blood and showed that they work in pigs. They also designed sensors that can respond to a molecule that is a marker of inflammation. Wireless communication. In the past decades, synthetic biologists have made great strides in engineering bacteria to respond to stimuli such as environmental pollutants or markers of disease. These bacteria can be designed to produce output such as light when they detect the target stimulus, but specialized lab equipment is usually required to measure this response. And to make these bacteria more useful for real-world applications, the MIT team decided to combine them with an electronic chip that could translate the bacteria response into a wireless signal. Our idea was to package bacterial cells inside a device. The device would be trapped and go along for the ride as the device passes through the stomach. For their initial demonstration, the researchers focused on bleeding in the GI tract. They engineered a probiotic strain of E. coli to express a genetic circuit that causes the bacteria to emit light when they encounter heat. They placed the bacteria into 4 wells on their custom-designed sensor covered by a semi-permeable membrane that allows small molecules from the surrounding environment to diffuse through. Underneath each well is a phototransistor that can measure the amount of light produced by the bacterial cells and relay the information to a microprocessor that sends a wireless signal to a nearby computer or smartphone. The researchers also built an Android app that can be used to analyze the data. The sensor, which is a cylinder about 1.5 inches long, requires about 13 microwatts of power. The researchers equipped the sensor with a 2.7 volt battery, which they estimate could power the device for 1.5 months of continuous use. They say it could also be powered by a voltaic cell sustained by acidic fluids in the stomach using technology that have previously developed. The focus of this work is on system design and integration to combine the power of bacterial sensing with ultra-low power circuits to realize important health sensing applications. Diagnosing disease. The researchers tested the ingestible sensor in pigs and showed that it could correctly determine whether any blood was present in the stomach. They anticipate that this type of sensor could be either deployed for one-time use or designed to remain in the digestive tract for several days or weeks, sending continuous signals. Currently, if patients are suspected to be bleeding from gastric ulcer, they have to undergo an endoscopy to diagnose the problem, which often requires the patient to be sedated. The goal with this sensor is that you would be able to circumvent an unnecessary procedure by just ingesting the capsule, and within the relatively short period of time you would know whether or not there was a bleeding event. To help move the technology toward patient use, the researchers plan to reduce the size of the sensor and to study how long the bacterial cells can survive in the digestive tract. They also hope to develop sensors for gastrointestinal conditions other than bleeding. In the science paper, the researchers adapted previously described sensor for 2 other molecules which they have not yet tested in animals. One of the sensors detected a sulfur containing ion called thiosulfate, which is linked to inflammation and could be used to monitor patients with Crohn's disease or other inflammatory conditions. The other detects a bacteria signaling molecule called AHL, which can serve as a marker for gastrointestinal infections because different types of bacteria produce slightly different versions of the molecule. Most of the work we did in the paper was related to blood, but conceivably you could engineer bacteria to sense anything and produce light in response to that. Anyone who is trying to engineer bacteria to sense a molecule related to disease could slot it into one of those wells, and it would be ready to go. The researchers say that the sensors could also be designed to carry multiple strains of bacteria, allowing them to diagnose a variety of conditions. Right now we have 4 detection sites, but if you could extend it to 16 or 256, then you could have multiple different types of cells and be able to read them all out in parallel, enabling more high-throughput screening. Good day, this is Professor Will, CBMD, PhD. Today, Saturday, May 26, 2018. It is 10:01 a.m. Eastern Standard Time. Science News. By forming clots and tumors, immune cells aids lung cancer spread. Date, May 24, 2018. Source, UNC Comprehensive Cancer Center. Summary. Researchers report that for a particular subset of cancer tumors, there is a high prevalence of immune cells called inflammatory monocytes. These immune cells, which normally help to build clotting scaffolds to promote wound healing, also make it possible for tumor cells to migrate and spread to other parts of the body. In the journal Nature Communications, researchers report for a particular subset of lung cancer tumors, there is a high prevalence of immune cells called inflammatory monocytes. These immune cells, which normally help to build clotting scaffolds to promote wound healing, also make it possible for tumor cells to migrate and spread to other parts of the body. The way that these immune cells promote lung cancer metastasis was very unexpected. They produce a large amount of a factor that leads to clot in the tumor, which the tumor cells can latch onto and climb across and spread into the body, said UNC Assistant Professor, UNC School of Medicine, Division of Hematology and Oncology, and the study's corresponding author. Our goal is to use this information to teach the cancer wounds to heal themselves. Previous studies have classified lung squamous carcinoma, which accounts for about 30% of lung cancers, into 4 different types based on their biological and molecular characteristics. In the new study, found that lung squamous carcinoma could be reclassified into just two different categories based on whether they showed a high presence of inflammatory monocytes. They used expression of the CD14 gene as a biomarker to show that high presence of this immune cell was linked to poor survival. It is important to understand that while there is so much focus on activating parts of the immune system to attack cancer, there is also a Jekyll and Hyde process going on in most tumors. There are immune cells we want to activate, but there are other immune cells we want to turn off. The researchers used newly developed laboratory models of lung squamous carcinoma to study the role of the inflammatory monocytes. The tumors make a signal called CCL2, which helps to recruit inflammatory monocytes. These immune cells then release a clotting factor, factor X111A, which creates a fibrin scaffold that tumor cells climb across and then travel to distant organs. These results shed new light on the tumor and microenvironment functioning and potentially may lead to new approaches for targeting the metastasis of this extremely aggressive disease. By genetically modifying the expression of CCL2 in the metastasis model developed in their laboratory, they found low expression was linked to reduced metastasis, while high expression was linked to enhanced metastasis features. They also demonstrated that the presence of a clot made it easier for cancer cells to move and migrate. Also, when looking at tumor samples from patients, they found that tumors with high amounts of the fibrin cross-linking was associated with the increased risk of the tumor spreading. The tumor researchers were able to reduce metastasis by using compound that blocks CCR2, a receptor on the surface of the inflammatory monocytes. They saw a significant decrease in lung metastasis. They want to continue investigating this strategy and potentially other ways to preventing clotting within tumors to see if it prevents metastasis from initiating and whether it can stall the process once it has begun. We want to make progress for patients with lung squamous carcinoma and expand their therapeutic options available to these patients. The more we understand the progression of the disease, including how metastasis occurs, the more we'll be able to understand how we can regress this disease or just keep it in check. We believe there are ways we can teach these tumors how to heal. Good day, this is Professor Will, CBMD, PhD. Today is Saturday, May 26, 2018. It is 10:22 a.m. Eastern Standard Time. 3D printed sugar offers sweet solution for tissue engineering device manufacturing. Date May 26, 2018. Source University of Illinois. Summary: engineers built a 3D printer that offers a sweet solution to making detailed structures that commercial 3D printers can't. Rather than a layer upon layer solid shell, it produces a delicate network of thin ribbons of hardened isomalt, the type of sugar alcohol used to make throat lozenge, the water-soluble, biodegradable classy sugar. Structures have multiple applications to biomedical engineering, cancer research, and device manufacturing. And the water-soluble, biodegradable glassy sugar structures have multiple applications in biomedical engineering, cancer research, and device manufacturing. This is a great way to create shapes around which we can pattern soft material or grow cells and tissue. Then the scaffold dissolves away. For example, one possible application is to grow tissue or study tumors in the lab. Cell cultures are usually done on flat dishes. That gives us some characteristic of the cells, but it is not a very dynamic way to look at how a system actually functions in the body. In the body there are well-defined shapes, and shape and function are very closely related. In a paper published in the Journal of Additive Manufacturing, the research group described the materials and mechanics of free-form isomalt printing. Free-form means that as the nozzle moves through space, the melted material hardens, leaving a sturdy structure behind like drawing a medallion. Other types of sugar printing have been previously explored but had problems with the sugar burning or crystallizing. The sugar alcohol isomalt could work for printing applications and less prone for burning or crystallization, and they had to build a printer that would have the right combination of mechanical details to print stable isomalt structures, the right temperature pressure to extrude it from the nozzle, diameter of the nozzle, and speed to move it so it prints smoothly, but then hardens into a stable structure. After the material and the mechanics, the third component was computer science. You have a design of a thing you want to make. How do you tell the printer to make it? How do you figure out the sequence to print all these intersecting filaments so it doesn't collapse? One advantage such free-form structures hold is their ability to make thin tubes with circular cross-sections, something not possible with conventional polymer 3D printing. When the sugar dissolves, it leaves a series of connected cylindrical tubes and tunnels that can be used like blood vessels, to transport nutrients in tissue or to create channels in microfluidic devices. Another advantage is the ability to precisely control the mechanical properties of each part of the structure by making slight changes in the printer parameters. For example, we printed a bunny. We could, in principle, change the mechanical properties of the tail of the bunny to be different from the back of the bunny, and yet to be different from the ears. This is very important biologically in layer-by-layer printing. You have the same material and you are depositing the same amount, so it is very difficult to adjust the mechanical properties. Using scaffolds and a variety of microfluidic devices and cell cultures, and it is working to develop coating for the scaffolds to control how quickly these dissolve. The additive manufacturing paper is part of the series of publications based on thesis work that details how to build the printer and the planning algorithms necessary to operate it, as the researchers hope that others can use their models to build printers and explore various applications for isomold structure. This printer is an example of engineering that has long-term implications for biological research. This is fundamental engineering coming together with material science and computer science to make a useful device for biomedical applications. Good day, this is Professor Will, CBMD, PhD. Today's Saturday, May 26, 2018. It is 10:37 a.m. Eastern Standard Time. Painless real-time proteomics may at one day speed up cancer surgery. Date, May 23, 2018. Source, American Society for Biochemistry and Molecular Biology. Summary, researchers have developed a matrix-assisted ion source for mass spectrometry that can liberate lipids and metabolites from the skin without causing pain. Now they have optimized protein measurements using this device. The device can be used to differentiate normal from cancerous tissues. Better surgery is associated with better prognosis and higher survival. The laboratory has worked for several years on a device they call the spider mask that will enable surgeons to look for markers of cancer in a living patient's tissue during an operation. In an article of Molecular Cellular Proteomics, the team reports on an important step toward finding protein biomarkers during surgery. Surgery to remove a primary tumor involves a wait after the tumor and tumor-healthy surrounding tissue are removed. The surgical team pause while a pathologist checks the tissue margins under a microscope. Although this process is important for preventing recurrence of the cancer, it can add up to 45 risky minutes under anesthesia. We think that it is possible to open up the way in vivo, real-time proteomics, which could help surgeons find stray cancer cells faster, perhaps even as they make incisions. Devices use mass spectrometry, which measures the mass of molecules from complex mixtures, but turning an in vivo tissue samples into gas phase ions for measurements can be a challenge. Until now, no one knew how to extract ions from living tissues without doing harm. Rifing on MALDI, an ionization strategy that uses a carrier molecule mixed with the analyte of interest, they decided to use the water that makes up the majority of human tissue as a carrier to produce a water-assisted laser deabsorption ionization, or WALDI. If they could excite the water in a tiny area, it should vaporize, taking ionized organic molecules with it. It was an idea at the beginning, and many people thought it wouldn't work. Finally, we have it working beautifully. The team built a pulse laser excitation device turned to heat water precisely by causing vibration in oxygen and hydrogen bond. In a 2016 paper, they described using this laser to ionize the outermost layer of tissue penetrating less than 1/20th of a millimeter. The human volunteers reported a slight tingling sensation, but the ions that appeared were mostly small molecules and lipids, which are more apt than proteins to adopt a negative charge. The team hoped to measure proteins as well. In this new paper, colleagues report that they have cracked the protein puzzle by using a more sensitive mass spectrometer and looking for positivity instead of negatively charged ions. They found peaks representing purified proteins they had introduced into a cow liver sample. Now that they know the proteins are detectable, the next step will be finding ways to amplify the protein signal over more abundant lipids and metabolites. In the meantime, the device is already in use for four-legged patients. Comparing biopsies from pet dogs with sarcoma to healthy tissues, the team developed a lipidomics and metabolics-based classification system to robustly identify healthy, necrotic, and cancerous tissues. Soon they will introduce a prototype into veterinary operating room. If it is successful there, team hopes to reach human clinics, improving tumor removal surgery to give patients better health outcomes.